Hello, everyone. Hope you're having a wonderful day. So today I'll be going over Casanova DB, which is the core model, a tool that we used in our paper. A learned score function improves the power of mass spectrometry database search. So I'll jump right into the background. A lot of this you might already know, but hopefully it should be good for you. How do I do this? Okay, got it. All right, wonderful. So let's start off by going over a very high level example of like a tandem mass spectrometry workflow. So you have samples from some organism, you take the proteins from that, digest it into peptides, usually between 6 to 50 amino acids long. You shoot that into the magic mass spectrometer and out comes an MS2 spectrum. I like to think of this as a peptide fingerprint. This piece of data, it's associated with a peptide, and you can use this information to quantify what exactly was in your sample to begin with, which has a plethora of uses, many of which are obviously not on the slide. But as most of you know, there's like a lot of reasons you want to figure out what's exactly in that animal soup you had at the beginning there. So moving on, a core problem within this workflow is actually figuring out which peptide generated that spectrum, because it doesn't come out of the machine with a nice little label on it. So how that is usually solved is through database search. So we start off with the spectrum that comes off the machine. And we also have a knowledge about the proteome of the organism that we actually put through the mass spectrometer. And using that proteome, we generate some candidates, which are chosen with their mass to charge ratio within a certain window of the precursor for that spectrum. So here are some examples, like from this organism proteome, some peptides that came out as candidates, DLV, QVS, and evil peptide K. So what we'll do next is, after this little like smart filtering we've done, is score them further to figure out which of these peptides most likely generated this spectrum. And this is kind of the core of what we're working on right now, the score function. So what exactly assigns these numbers to these peptide spectrum pairs, right? And so when you've done all this, essentially you can say that little green peptide I've selected there, that is what we believe has generated that spectrum. So how do we make sure that we're matching to the actual correct generating peptide and not something else that's in this candidate list? We throw in a bunch of decoys in there, which we know are decoys. I've marked them in red. And the idea is, if we have a spectrum that matches to one of those decoys, we know something went wrong. And we can add 1 to our estimate of how many incorrect target matches we have made. I've illustrated this on the next slide. A lot of colors, sorry. So we're going to start off with just like the labels here. We have the decoys are the red ones. We actually can know where those are in our database. And then we have the wrong targets and the correct targets. We cannot necessarily distinguish those. But what we can do is figure out like where we can place the score threshold so that we're pretty confident about the peptides we accept within our workflow. If we move this over to the right, increase the score threshold, we will get, we'll identify only a few spectrum, but we'll be very, very confident about what we identify. Move it to the left, we can accept a lot more, more comprehensive analyses downstream, more interesting results, but there's a high likelihood that a lot of the stuff in there is going to be bogus. What we would like to do is find a nice sweet spot where we have a low amount of false discoveries. Usually it's around 1%. And as you can see here, the amount of decoys we have in that database estimates the number of wrong target matches that we have. So this is how I like to think of target decoy competition, using it to control the false discovery rate. So I'll just be using those terms throughout this talk. So with all this being said, one question that we had is kind of like, we know that a good score function is crucial to actually making sure we do these matches properly, and we want the numbers we assign to be obviously higher for the targets and much lower for the decoys. So how do we create a score function that leverages the huge amount of like tandem mass spectrometry data out there on the internet to increase the separation power further? So like, can we actually do better than just some simple algorithm? Can we like use data to make this better? And we started looking at like, how do we exactly learn a score function, right? Now, small tangent, but coming back to the main point, is this tool that the Nova Lab has developed called Casanova. It solves the problem of spectrum identification in a de novo setting, so without a database. what it does is you give Casanova this deep learning model, the spectrum information, and out comes a peptide on the other side. No database needed, just add water or whatever, right? So the idea behind Casanova is that despite it not being explicitly trained on scoring peptide and spectrum, it has an understanding within itself, understanding in a very loose term, of the relationship between spectra and peptides, right? So our hypothesis is that we can look inside Casanova, kind of pick it apart, query it, and leverage its learned relationship between peptides and spectrum and use that as a data-driven score function. So it's pretty interesting, applying like a de novo tool to a database search setting. So yeah, that should be all the background that's necessary for this. So we went over like a little bit of like the exact like peptide spectrum matching problem, FDR and Casanova. Now I'll be going over the implementation of our tool, like how exactly we did this and like why it would score peptide and spectrum with greater separation power. So moving on. I won't be going over a deep dive into transformer models or anything, despite Casanova is a transformer model that does autoregressive next token prediction. A lot of words there, and there's a lot of words on the image as well, but I'm going to highlight the parts that are important. So how Casanova actually makes de novo predictions for peptides is, say we're in the middle of predicting a sequence, right? And it has so far predicted PEP-TID. So it takes into consideration the spectrum information as well as those previous amino acids it has predicted. And for every single amino acid in its vocabulary, I have a couple here, it generates a score. So it says, I think A doesn't really belong in the next position. I also don't think C belongs in the next position, definitely not D, but I do think E belongs there, right? So that's going to be given to us with like the highest number there in that little list. And what we're going to do is say, all right, you think E belongs there, wonderful. We're going to put it there and then keep predicting until you tell us to stop. So that's generally how autoregressive next token prediction works for these transformer models and for Casanova. So maybe you're seeing how we can start extracting insights here for a peptide spectrum pair. So what Casanova DB does, and this is getting into like exactly how the tool works, is we essentially query Casanova. We use it for a different purpose than it was originally designed. Instead of letting it do the predictions itself, we're asking it questions about what it thinks about this amino acid belonging here for this spectrum information. So I'll walk through a couple examples. So say we're working on a target right now, and we ask it a question, hey, given the spectrum information and a start token, what do you think about W being in that first position on this peptide? And it'll give us a score. And it'll say, oh yeah, I really think that belongs there. And it doesn't know this, but say that's the generating peptide. And it says, all right, I think that score is really high. Great, I would put that there. And it gives us that number. And then we ask it, all right, given the start token and that W that we just gave you, what do you think about this next token? And we say, or what do you think about N? And it says, oh yeah, I think that also belongs there. I'm going to give it a score of 0.95. And then it's essentially rinse and repeat for the rest of the amino acids in that target sequence. And we get this list of numbers that essentially tells us for each amino acid, how confident exactly is Casanova that that amino acid belongs there in that position, given that spectrum information, right? So that's how Casanova would operate on a target. Now, if we look at how it works on a decoy, and notice the first two letters are the exact same between the targets and the decoy, just for illustrative purposes. We ask it about W, same thing, right? Because we use the same spectra. We ask about N, same thing, right? Same information, exact same thing. So it's deterministic in this case. But then we ask it about the letters that deviate, the G and the K. And Casanova does not think those amino acids belong in those positions. And so it gives us these low numbers as a result. And this happens because in a de novo setting, Casanova understands that certain amino acids belong in certain places given a spectrum, and we're asking it to score something that really does not belong there because it comes from a decoy, and it recognizes that and spits out a low number, right? And we take these low numbers and aggregate them into a list just like we did before. A small note, the difference between autoregressive decoding and what we're doing here is even though Casanova would not put a G in that third position, it would most likely put a Q because that's the generating peptide. We still make it consider the G in that case, and that's called teacher forcing, because we want to know what it thinks about the entire peptide, one after the other. What we can do with this now is just take these lists, these aggregates of these scores, and just take the geometric mean of them. And what that gives us is akin to, not akin, it's exactly database search results. So we have for each peptide spectrum pair a score that goes along with it. And that score, we can just take the highest scoring peptide and say this is generating peptide for this spectrum. Wonderful. So now we've taken a de novo tool and applied it to a database search setting, and it works great, which would be the next section of the talk. I'm going to go over the experiments we ran, some FDR curves, and some other orthogonal things we've done to determine that CasinoDB is giving us some pretty useful information. So here are some benchmarking decisions we made. We took MSMS runs of 3 different organisms: human, E. coli, and yeast data. All of these runs were done after the training data for Casanova was created, so there is no chance for leakage there at all. And we also had 2 of the organisms, E. coli and yeast, Casanova has not seen before either. So it's trained on human data, but we're also testing on 2 entirely different organisms. We also chose 3 widely used search engines to compare against, although we're not really comparing against the search engines, we're comparing against the score function. So we use Tide, which has the X-score score function, Sage, which uses hyperscore, and Andromeda, which uses the Andromeda score function. So these are the comparisons, just setting up like the experiments that I'm going to show in the next couple slides. And we also made sure to try and employ the exact same parameter settings for all these search engines, so that really what we're comparing is Casanova DB as a score function versus these other score functions, not necessarily a database search. So here's how we interpret these FDR curves we got. We see this red dashed line there, which is placed at 1% FDR, and where it intersects those curves is how many peptides you can safely accept at 1% FDR. So as you can see here, Casanova DB being that blue line, it goes over the other 3, which would be Tide, Sage, and Andromeda. And what that means is at 1% FDR, we can accept a lot more peptides safely, essentially saying that we're not going to have so many false discoveries. And we see that this is true for all the species across the board. We see that like Casanova DB accepts a lot more peptides at 1% FDR as compared to the other score functions, meaning that yes, we were successful in creating a learned score function that has higher separation power between targets and decoys. We also threw in Percolator into the mix because that's a very common tool to use within mass spec workflows. And we use a very minimal feature set, simply just score, charge, and the mass to charge ratio. That's it. That's all that went to Percolator. And our advantage grew even further for a lot of these organisms and search functions that we use, score functions, sorry. And what this tells us is that with a little bit of alignment, Casanova DB can work even better. And the advantage is not taken away by using something like Percolator. We still do very much have an advantage over the other score functions. And this is also true, although I didn't show them on these slides, for the other organisms as well. And here is kind of a summary table, just numerically, to see how much on average more peptides we accept at 1% FDR as compared to the other score functions. So you can see like for the right side with percent increase after Percolator, we sit between 40 to around 70% increase in the number of the spectrum identifications, or 1% FDR. And now we want to make sure that these identifications we have, they're not bogus. And we want to make sure that obviously for these tools that have worked great in the past, for other experiments, that we are discovering the same things as them. So we use an upset plot to compare which peptides we discovered between like all the different search engines. And we found that of the around like 37,000 peptides that we were able to accept at 1% FDR, in this case for the human, we only missed 54 peptides, in the sense that the other 3 score functions total only found 54 peptides that we didn't find. Essentially, not that much. So the little graphic I have there should illustrate the idea that this upset plot is trying to communicate, is that we find almost like a superset of the peptides that the other score functions are able to find. And this is not shown on the slides as well, but we did another experiment where we checked at 10% FDR what the other score functions found. And it would, a lot of those peptides we had found at 1% FDR, meaning that we are getting better separation power through the use of Casanova DB. So that was the experiment section. We went over the 3 different organisms we used, the FDR curves related, and also this orthogonal experiment showing the overlap in identified peptides. And where does this leave us? So the basic implementation is fully available for everyone to use. It's on the Casanova repository in this little branch over there, waiting on a pull request. Make sure it's so to get it into like the main branch. And after that, it'll be just part of the Casanova install normally. You give it an MGF, a FASTA, and you get MZTab output with a score for every single peptide spectrum pair. And as I mentioned earlier, it should be merged into the main branch soon. TLDR, it's easy to use. I'm totally willing to help troubleshoot over email. Got not much else to do this summer, so just hit me up. And one last thing I'm going to go over is the improvements to come. So first of all, we want to make sure this is really well documented so anyone can use this to either benchmark against or just use for their own purposes. And in the longer timeline, we want to do model distillation on Casanova DB, kind of prune or trim the fat, and then so we can improve the speed and the runtime of this so you can do a lot more spectra in one run. And also we want to add more comprehensive database search features into the tool. We have some pretty basic ones right now, but those can obviously be expanded with a little bit of software engineering. So with that, thank you so much to the lab. I love you guys. You guys helped me so much and been so patient with my like millions of questions I always ask. So it's been a great time at the Noble Lab. And with that, I would like to open the floor for any questions.
Again, great talk. I have a question regarding your percolator experiments. You said that in addition to the Casanova score, you only use charge and M over Z of the precursor. And I believe there was like a 20% gain of identifications. Does that, is it fair to make the statement that that means that Casanova has a bias when it comes to charges and the corresponding score? And would it be possible to eliminate that bias by training Casanova differently, such that percolator doesn't give that 20% increase anymore?
Yes, so it's kind of illustrated here. If you see on the left before percolator, our curve is actually shifted over to the right a little bit. And the reason that is because Casanova is actually just inherently biased towards shorter peptides, just the way we did the scoring. It's not on here as well, but when we first saw those curves, we did like an analysis, like what are we messing up earlier on with? And they're all shorter peptides, right? So percolator fixes that. So while I can't really say that it's like the bias is towards like charge or anything, we have done investigations and Casanova is definitely biased towards shorter peptides. That could definitely be trained out, I believe, because the Casanova was trained on a totally different like domain, de novo sequencing, and without any fine tuning at all. There's no fine tuning in this whole experiment. We're able to get this additional separation power. So if someone was to go ahead and fine tune Casanova specifically for database search, I do believe this could be eliminated.
Thanks.
Hi, thank you for fantastic talk. Very educational, I think. So thank you for that. I had a question because like what I found is the most interesting of Casanova DB is that for the first time you don't necessarily need to have one score for the whole peptide. You have your residue precision. So I was wondering whether instead of collapsing into one scoring function and one number, you could also output the array of scores for each amino acid, because then you get that sensitivity to then you don't need your proteogenomics database. You can just look at which residue you may have wrong. Maybe like 99% of your sequence is correct, and you get one residue that's wrong, and then you can even think of PTMs. So I was wondering if you have tried that or if it's in the future where.
Yeah, so the MZTab output does have those scores in a list aggregated. So if you want to run any experiments, you could just run the tool, and then there's a column that just has those scores right there. Personally, the only thing we did with that list of scores was collapse it into like a total score so we could do some FDR control tests. But yeah, I'd totally be curious to talk about like what we could do with that after.
Yeah, well done. Thank you.
Hi, very interesting. Just can you comment briefly on the practicalities of memory requirements and runtime for this tool?
For sure. So based on my quite rough experiments before I made my way over here, I like Control C, Control V to huge MGF and just ran it through. And just projecting, it should take around for 1 million PSMs, would assume that it would take around like less than 30 minutes or so. And I wish I could pull it up right now. I should have put it on the slides. But I believe it's in like 0.0001 seconds per spectra or per spectropeptide pair. I do think it is slower than most methods. Obviously, it uses deep learning, and you need a GPU to run this at the speeds you do. But the model distillation part I was talking about earlier, part of speeding it up is so that we can run it on a CPU with, hopefully enough speed such that you can run a decently comprehensive analysis. But yeah, feel free to talk to me after. I'd love to think about ways to do that.